Today, an FDA approval in acute myeloid leukemia, a priority review designation in prostate cancer, a new drug application in prostate cancer, and European recommendations in ovarian cancer, breast cancer, and cutaneous squamous cell carcinoma. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved a supplemental new drug application for single-agent evosidenib for the first-line treatment of adult patients with IDH1 mutant acute myeloid leukemia as detected by an FDA-approved test who are at least 75 years old or are ineligible to receive intensive chemotherapy. The approval is based on findings from a phase 1 trial in which evosidenib induced a 28.6% complete response rate and a CR plus CR with partial hematologic recovery rate of 42.9%. The median durations of CR and CR plus CRh were not estimable, and 41.7% of patients who achieved CR or CRh remain on evosidenib as of the data cutoff. Results also showed that 58.3% of patients who achieved a CR or CRh were in remission at 1 year after receiving evosidenib. The median time to CR/CRh was 2.8 months. Also, among 17 patients who were dependent on red blood cell and/or platelet transfusions at baseline, 41.2% were independent of RBC and such transfusions during any 56-day post-baseline period. Moreover, of the 11 patients who were independent of both RBC and platelet transfusions at baseline, 54.5% of them remained transfusion independent during the similar period. The agency's decision occurred more than a month before the action date, which was June 21, 2019. The FDA has granted a priority review designation to a new drug application for darolutamide for use as a treatment for patients with non-metastatic castration-resistant prostate cancer. The NDA is based on data from the phase 3 ARAMIS trial in which the investigational nonsteroidal androgen receptor antagonist was associated with a 59% reduction in the risk of metastases or death compared with placebo in this patient population. Bayer, which is manufacturing darolutamide in collaboration with Orion Corporation, stated in a press release that darolutamide has also been granted fast-track designation in this setting. At a median follow-up of 17.9 months, results showed the median MFS was 40.4 months with darolutamide versus 18.4 months in the placebo arm. Additionally, the MFS benefit was observed across patient subgroups, including those for baseline prostate-specific antigen doubling time, use of bone-targeting agents, Gleason score, age, and ECOG performance status. The 3-year rates of OS were 83% in the darolutamide arm versus 73% with placebo, translating to a 29% reduction in the risk of death. Moreover, the median OS was not yet reached in either arm. Also in prostate cancer, a supplemental new drug application has been submitted to the FDA for the approval of apalutamide for use as a treatment of patients with metastatic castration-sensitive prostate cancer. The application is based on data from the phase 3 TITAN trial, which showed that apalutamide plus androgen deprivation therapy significantly improved radiographic progression-free survival and overall survival compared with placebo and ADT in this patient population. Full findings from the study will be presented at the 2019 ASCO annual meeting. In January 2019, it was announced that due to these data from a pre-planned analysis, along with a recommendation by an independent data monitoring committee, the study had been unblinded. Based on the positive results favoring apalutamide, it was recommended that patients on the placebo and ADT arm be permitted to cross over to receive apalutamide. Patients will continue to be followed for OS and long-term safety as part of the TITAN trial. The sNDA is being reviewed as part of the FDA's Real-Time Oncology Review Program, which is designed to have a more efficient review process to make therapies more quickly available to patients. The FDA initially approved apalutamide for the treatment of patients with non-metastatic castration-resistant prostate cancer. The European Medicines Agency's Committee for Medicinal Products for Human Use made a handful of positive opinions this past week, recommending drugs for approval in breast cancer, ovarian cancer, and cutaneous squamous cell carcinoma. First, the committee backed approval of talazoparib for adult patients with HER2-negative, locally advanced or metastatic breast cancer harboring germline BRCA1/2 mutations. If approved, the PARP inhibitor's indication would stipulate that eligible patients must have prior treatment with an anthracycline and/or a taxane in the neoadjuvant or adjuvant locally advanced or metastatic setting. Eligible patients with HR-positive disease also must first have received endocrine-based therapy. The recommendation is based on data from the Phase 3 EMBRCA trial, in which talazoparib reduced the risk of disease progression or death by 46% versus chemotherapy in patients with BRCA-positive advanced breast cancer. In ovarian cancer, olaparib was recommended for approval as a frontline maintenance treatment for patients with BRCA-mutant advanced high-grade epithelial ovarian, fallopian tube, or primary peritoneal cancer who are in complete or partial response following first-line platinum-based chemotherapy. The positive opinion is based on findings from the Phase 3 SOLO-1 trial, in which olaparib led to a 70% reduction in the risk of disease progression or death following response to platinum-based chemotherapy compared to placebo. And in cutaneous squamous cell carcinoma, the committee issued a positive opinion recommending approval of cemiplimab for the treatment of adult patients with metastatic or locally advanced cutaneous squamous cell carcinoma who are not eligible for curative surgery or curative radiation. The recommendation is based on a combined analysis of data from the Phase 2 EMPOWER CSCC1 trial and 2 advanced CSCC expansion cohorts from a Phase 1 trial of patients with metastatic and locally advanced CSCC. Across the entire population, the overall response rate at a median follow-up of 8.9 months was 47%, the complete response rate was 4%, and the partial response rate was 44%. The duration of response ranged from 1 month to over 15 months. Moreover, 61% of patients had a duration of response of at least 6 months. This week, we sat down with Dr. Vicki Macker of Memorial Sloan Kettering Cancer Center to discuss evolving treatments for advanced endometrial cancer.
I'll start by discussing just the incidence and mortality of endometrial cancer, and also sort of cover the landscape in terms of therapy for patients in the adjuvant disease setting and the advanced disease setting in this disease as well. Endometrial cancer is the most common gynecologic malignancy in the United States, and alarmingly, the incidence and mortality associated with this cancer are actually sharply on the rise. There are a number of factors that are felt to be responsible for this alarming trend in this disease. Chief amongst them is obesity. So obesity causes a number of perturbations that can directly promote tumorigenesis. Obesity is now reaching epidemic proportions in this country. Among the perturbations that can occur as a result of obesity include chronic inflammation that can be peritumoral and also can occur distantly. That can also lead to hyperglycemia, hyperinsulinemia, insulin resistance, abnormalities in the insulin IGF-1 signaling pathway that can directly lead to activation of the PI3 kinase AKT mTOR pathway that can promote tumorigenesis. Obesity can also lead to adipokine pathophysiology alterations and really other changes that can promote this disease. So this is a very large problem in this cancer specifically. However, thankfully, most patients with this disease are still diagnosed with early stage disease. However, approximately 21% of patients will have regional spread of disease to either pelvic lymph nodes or adjacent structures, and approximately 9% of patients will have distant metastases at time of diagnosis. In terms of the management of endometrial cancer patients, so the initial step is surgical resection for the vast majority of patients, and that surgical resection and proper surgical staging is very important because the stage informs treatment. Other factors that are very important in this disease in terms of informing treatment include the histology of the disease and also the molecular phenotype of this cancer. So with regards to upfront treatment, adjuvant therapy, the goal is to reduce the chance of recurrence. And it is those factors, again, the histology, the stage, and the molecular phenotype, along with adverse risk factors that really inform adjuvant therapy for patients. Adverse risk factors in this disease do include aspects of the tumor, such as high grade, advanced age of the patient, lymphovascular invasion, deep myometrial invasion, and then other factors such as lower uterine segment involvement, for example. With these factors, we determine whether patients are high risk or of lesser risk. Patients that are considered high risk include what we classically think of as type 2 endometrial cancers. So these are the more rare subtypes of uterine cancers. They include serous, clear cell, carcinosarcoma, and other rare subtypes. Other patients that are considered high risk include the grade 3 endometrioid adenocarcinomas that have deep myometrial invasion, certainly the stage 3, stage 4A patients, regardless of the histology. And the reason these patients are high risk is that they really have aggressive behavior and can lead to early recurrences. So for these patients, patients, the general approach is multimodality therapy, even for the very early stage patients. For patients that are advanced stage and are deemed unresectable, the usual approach is to offer chemotherapy. The role of radiation therapy in these patients is individualized and is based on the bulk of tumor and also location of tumor. The additional factor that I think is very important in endometrial cancers is the molecular phenotype, which I keep alluding to. And so in 2013, the TCGA Endeavor really refined our understanding of endometrial cancers and really defined 4 subtypes of endometrial cancers that are associated with different progression-free survival. So these 4 subtypes include the following. The first are what we call the poly-ultramutated subtype. This subtype is associated with hotspot mutations in the catalytic subunit of DNA polymerase epsilon. This subgroup includes the endometrioid adenocarcinoma subtypes as far as histology is concerned. More than 50% of these patients are the grade 3 endometrioid adenocarcinomas. And this subtype actually has the best survival. In fact, it comprises about 1% of the recurrent disease patient population. The other next subtype is the microsatellite instability hypermutated subtype. This subtype is also comprised of the endometrioid adenocarcinomas. It is associated with mutations in the DNA mismatch repair genes that are involved in DNA replication and repair. This subtype comprises about 25% of patients in the recurrent disease patient population. So again, overall, this subtype tends to do well. The remaining subtypes are what we call copy number low and copy number high subtypes. So the copy number low are comprised of the low-grade endometrioid adenocarcinomas. These are the grade 1, grade 2 subtypes. The copy number high group are the serous or serous-like groups. So this subtype has very high number of copy number alterations, has mutations in TP53, and the molecular landscape of this particular subtype overlaps with basal breast cancers and serous cancers of ovarian etiology. But there are notable differences. So for example, about 25% of this subgroup has amplifications in ERBB2, which is a target that is relevant in the advanced disease population. The copy number high or serous-like group is really associated with the worst progression-free survival. And so this is a very aggressive subtype that we worry about in the recurrent disease population.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.